Hi, everyone. Welcome to today's webinar. We're just going to wait a minute for everybody to get logged on, and then we'll get started. Hello, everyone. I'm Kirsten Wakefield, Stakeholder Engagement Specialist for Maricus, and together with Dr. Janet Reamer, the new Science Program Director for MARCO, we co-coordinate the Mid-Atlantic Coastal Acidification Network, MACAN. Today, we're excited to hear more about how the coupling of biological, biogeochemical, and economic models can help quantify impacts of acidification and other climate stressors on oyster growth and aquaculture economics in Chesapeake Bay. But before we get to the presentation, a quick look at our upcoming webinar series. Our final webinar in May will feature vulnerability assessments and adaptation planning for the Northeast Atlantic sea scallop fishery and the Chesapeake Bay oyster restoration and aquaculture industry, as well as a comparison of climate resilience and wild versus cultured broodstock of the eastern oyster. And registration information for that webinar will be coming out May 1st. We also recently transitioned our listserv to Constant Contact, so please check your email and your spam folders for the new format for our newsletter. And you'll be able to find us on Facebook and Instagram shortly as well. Today, we'd like to give a warm welcome to our featured speakers, Dr. Jeremy Testa and Matt Weber. Jeremy is an associate professor of estuarine systems ecology at the University of Maryland Center, Cooperative Eastern Shores Chesapeake Biological Laboratory. And Jeremy's research focuses on the processes of eutrophication, nutrient cycling, and coastal hypoxia and acidification. He applies his research to ecosystem management in a variety of ways, including participation in the seasonal Chesapeake Bay hypoxia forecasts, co-authorship of the Maryland Ocean Acidification Action Plan, and as an at-large appointee for the Chesapeake Bay Program Scientific and Technical Advisory Committee. Our second speaker today, Matt Weber, enjoys researching challenging topics in environmental economics to inform improved policy and management. His recent projects include the one he'll be presenting on today, as well as incorporating social vulnerability and U.S. flood risk reduction investments and the public value of wild salmon recovery in the Willamette watershed. Matt has been with the UMC's Chesapeake Biological Lab since 2017 and has over 20 years of experience in consulting with the government, the nonprofit sector, and academia. And today, Jeremy and Matt will discuss how their collaborative approach to modeling can be used to investigate the complex interactions of a variety of climate stressors on the economics of the Chesapeake Bay oyster industry and aquaculture. So we will hold all questions today until after the presentation, and all of our participants will be muted, so please ask your questions via the Q&A box at the bottom of your screen. Anthony Himes, our Sea Grant Ocean Acidification Fellow, will help moderate the Q&A discussion. And just a reminder that this webinar is being recorded and will be available on the MACAN website, midacan.org, shortly. So without further ado, let's turn it over to Jeremy.
Thank you. Thank you for that introduction, and thanks to MACAN for having us here. Can you confirm that you can see the slides okay?
Yeah, and Jeremy, thanks.
Great. All right, well, welcome everyone, and thanks in advance for listening. I'm going to describe with Matt kind of a summary of this project, somewhat ambitious project we took on to try to couple three different classes of models together to try to learn something about primarily when we set out the vulnerability of oyster aquaculture to acidification. But as things go in research, we ended up being interested in a slightly broader view of all of the different physical and biogeochemical factors that'll change in the future and how they kind of work together and with each other and against each other to drive outcomes for oysters. And so we'll try to sort of summarize that today. This project was part of a sort of multi-investigator team. So just to introduce those folks, all of whom can't be here, Ming Li at Horn Point Laboratory was part of the physical modeling and biogeochemical modeling group. I'm in the middle. Wei Jun Kai was working on aspects of the acidification chemistry. George Waldbusser on the bottom left was working on the oyster modeling and oyster physiology. Lisa Wanger was working with Matt on the economic modeling part of this, and Matt Weber, who will talk in 20 minutes or so, is on the bottom right. And so we came into this project interested in oysters, but sort of more broadly as oysters being, a valuable ecosystem and economic entity that was affected by acidification. And just to introduce for a moment, just the complication of investigating a problem like ocean acidification and estuaries, simply because of the complexities in how carbonate chemistry is affected by things happening in environments that are highly affected by riverine inflows versus influenced by the ocean. So just to illustrate that with this somewhat complex diagram, if we think about the factors that drive the potential for estuarine acidification, which is a term we can use, estuaries get the acidification effect through nearby oceans taking up carbon dioxide and therefore having pH be reduced and other chemical factors and exchanging with the estuary. And some estuaries, especially large ones, can take up carbon dioxide themselves and sort of acidify from the atmosphere. So that's kind of the traditional ocean acidification picture. But if we think about estuaries, they're kind of fundamentally driven by freshwater inputs, and those freshwater inputs can have really somewhat variable chemistry, often chemistry that involves high carbon dioxide concentrations and low carbonate saturation state, and often poorly buffered water. So the interannual changes in freshwater can have a large effect on acidification. Estuaries are extremely productive. So, primary production or photosynthesis in the water can take up carbon dioxide and elevate pH, whereas respiration in the bottom water, which breaks down organic material, produces CO2, leads to pH decline. Both those happen at really high rates and can affect the conditions. And then even the biota living in the estuary, including the oysters themselves, can have effect on the carbonate chemistry. And so we kind of had to reckon with this backdrop of complexity when we came to this topic of looking at Chesapeake Bay in the future. And I was sort of assuming I wouldn't have to make this point to this group, but we chose oysters as our organism of interest for a number of reasons. But in summary, there's a historic wild oyster population and harvest that went along with that had very large economic effects. That wild harvest has gone down over time, but still exists. And in some ways, to compensate for that loss of wild harvest, there's been an increase in aquaculture to grow oysters, especially in Virginia, but increasingly in Maryland as well, associated with some leniency in the regulations that happened about a decade ago or a little more. So there's a very strong economic reason to be interested in oysters and how they'll change in the future, but they're also important parts of the ecosystem in a diverse collection of ways. And so just understanding, their potential future has impacts for things like nutrient cycling and water quality. So the motivating questions that we came into in this project was, originally just thinking about how future climate in general would offer or alter the acidification picture in Chesapeake Bay, across Chesapeake Bay. Secondly, to consider how acidification and other environmental changes may impact the growth and survival of the eastern oyster, primarily in aquaculture settings, and then how those changes in oyster survival and growth, when considered in an aquaculture setting, would relate to thresholds and other constraints on the oyster aquaculture industry. And as Matt will show, and I won't say much more about now, we'll consider how, the potential, I guess, for the aquaculture industry to mitigate against acidification and other stressors, and what it might cost to do that, how it'll affect their bottom line. So this is a complex project. It includes a lot of different variables over a lot of different timescales. And so to take on this problem was really not something you could do with observations alone. So we sort of considered and did this project using a coupling of a bunch of different models. So just to describe those in general, we started with downscaled climate projections for the Chesapeake Bay region. And so in this talk, we'll be considering sea level rise, warming, changes in freshwater input associated with changes in precipitation, and of course, changes in atmospheric carbon dioxide. Those forces are used to drive a biogeochemical model that simulates, tens of thousands of cells in Chesapeake Bay and some of its tributaries. And we considered current and future conditions for salinity, temperature, pH, omega, this saturation state variable that controls or factors into the energetics of oysters. And we consider changes in food. And so, all of these variables are predicted to change in the future, as I'll show you in summary. And so just considering for one, temperature is predicted to increase in Chesapeake Bay. It already has increased in Chesapeake Bay. And so considering those kind of factors is what we're working on. And so we use that biogeochemical model to predict oyster growth in 2 decades, a contemporary decade centered around the 1990s, and a mid-century decade focused around the 2060s. And we used a bioenergetics model to simulate triploid oysters. I won't talk as much about the survival work that we did, but we used kind of baseline survival estimates that were available from agencies. And because salinity in the northern part of Chesapeake Bay and the low salinity parts is a really strong stressor for oysters, because salinity can get so low, sometimes for extended periods, we considered salinity-induced mortality from interannual variability in freshwater flow and how that might change in the future. And then we use those estimates of growth to support Oyster economic model, which Matt will talk about, which considers the behavior of growers. It considers the input budget for an aquaculture farm, and it considers economic outcomes, primarily the economic output of profit, thresholds in that profit, which are you making a profit or are you not? And something called value of information, which Matt will talk about a little, I think. Okay, so I kind of want to walk through each one of these models quickly before I go on to the other parts of the presentation. There's a lot of different climate models that have downscaled projections for the Chesapeake Bay region. For this particular talk, we just used one type of model that did one of these simulations. And just to give you an example, over the course of a year here, these models predict somewhere between a 1 to 2 degrees Celsius change and a seasonally varying change in river flow. So this particular model suggests increases in river flow in the winter, spring, and perhaps decreases in river flow in other parts of the year, especially the fall. And so these changes in climate have a big effect on the biogeochemistry of the bay. And so if we look at what, in summary, this is going to be relatively short, what changed in the model. What you're looking at here, I realize this is a lot of information at once, but on the left are each one of these panels is a map of Chesapeake Bay where the Susquehanna River comes in. At the right here, or at the left, I'm sorry, and then over here at the right is where the Atlantic Ocean is. And so you're looking at this for salinity, total alkalinity, dissolved inorganic carbon, and pH, all parts of what we call the carbonate system, dissolved oxygen, and then this omega aragonite, which really kind of is a metric of how vulnerable shell might be to acidification. And so we have the 90s here in the middle panel, we have the mid 21st century, the 2060s. And then maybe what's more interesting is here on the right, where we have the difference between those two periods, where it's the future minus the present. So if you see a positive number, that means there's an increase. So getting to the heart of what's relevant for the oyster, this model predicts an increase in salinity over time, about somewhere between 1 and 2 salinity units. And that's pretty sort of uniform across the bay. It predicts a decline in pH on the order of, say, 0.2 roughly pH units. And that decline is kind of focused in the lower part of the bay and in kind of the water column of the middle part of the bay. And then this aragonite saturation state declines, which would be associated in some ways with a pH decline. And that would indicate that for example, it may be somewhat harder for the organisms to sort of build shell around the environmental conditions. So if we think about some broader changes in some of the other variables across the places where we looked, and sort of emphasizing that this is a multi-stressor process, what you're looking at here now is kind of a summary of those changes at a bunch of different locations in Chesapeake Bay where I'm going to show you some model output for oyster growth. And so in this case, you're looking at an individual location derived from the model, from the left going from the upper bay to the lower bay. So if you look at salinity, for example, you can see there's a really low salinity site we modeled all the way down to a site right at the mouth of Chesapeake Bay. And the red bars are the future and the green bars are the 1990s. And so you can kind of see this sort of general pattern of warming, more salinity, and then, slightly more acidic conditions. So we use these predictions of what the future will look like to drive an oyster bioenergetics model, which we adapted from a number of published models out there. And although I can't go into detail about how we did all of this, I'm going to try to show you some of the detailed output in a couple of slides. And I kind of want to give you the basics of how this model works. So we first have the output from the numerical model that simulates all of the biogeochemistry and all the physical variables over time. And then we have an oyster, one single individual oyster that's growing in response to these variables. So the oysters get their food through filtration and ultimately ingestion, and that's a function of temperature, of chlorophyll A and particulate organic matter, or POM, that's the food, and then salinity. And we use sort of Chesapeake Bay oyster literature to derive some of these formulations. So for example, this is an example of a few different models of which we use kind of a synthesis of that relate filtration to salinity. So when salinity is less than 5, filtration really slows down. There's kind of an abrupt transition to basically when you get up to 10 and oysters are kind of filtering as fast as they could filter. There's similar types of formulations for temperature. And really the model was kind of linearly, ingestion was more or less linearly related to chlorophyll and PLM. So the other part of the term, that's the input to the bioenergetic calculation, but then there's excretion, and then there's metabolism like respiration. And so, for example, metabolism is a function of temperature. So when it gets warmer, their respiratory demands go up. And then the balance of these two leads to the potential for absorption and assimilation, basically what's left over for growth and reproduction. We didn't simulate reproduction because we focused on triploid oysters. But the way we considered the acidification effect was drawing on some of George Waldbusser, our colleagues' work on, how calcification rates changed with the saturation state index. And so effectively this worked to be kind of a tax on the net energy balance, with the idea that as conditions become more acidic, then oysters are going to have to spend more time or more energy, I should say, sort of reaching the status quo. And so there's much more to talk about there, but that's kind of where we ended up. And so one of the things I want to point out before I go any further is that there's a fair amount of limitations to this kind of model. We're really kind of simulating a condition that doesn't really consider disease. It doesn't consider genetic variation in the wide variety of oysters that could be out there. We don't consider larval effects, and it's kind of limited how we can deal with site specificity. We're kind of considering this bay-wide view, but farms are occurring along the shorelines in a lot of different environments, and our ability to capture all of that is limited. And so to just give you like one example of that, this is a picture of oysters from a farm we worked at out in front of the Chesapeake Biological Laboratory in the Patuxent River. And so you'll see these are oysters about ready for harvest. But if we look at a histogram of the length of those oysters, just in one bag, in one cage on this farm, so you can see that basically the majority of these oysters are sort of, are at sort of harvest readiness. There's a bunch that are a lot bigger and there's a bunch that are a lot smaller. So there's a lot of variability even within a bag, and we're trying to model this individual oyster. And so this is just a picture from another bag of oysters that were a lot smaller, and you can just see the variation that happens. So it's it's a complex topic and one we can only kind of begin to scratch the surface of, but we did what we could. Okay, so when we do every time we model, we have to consider trying to validate the model in some way. And so the way that we chose to validate the Oyster model, at least, was to use some growth measurements that were available for a farm that was in the Potomac and a couple of others from around the Bay, but we primarily used data from this Potomac farm because it was the best that we had. And so that's what these green dots here, that's shell weight and tissue weight, which are the variables coming out of the model over the course of about 5 years. I use 5 years here because we ran 5-year simulations under the same sort of conditions repeated for any given year. And then we reran 10 different years based on the 10 years that we had in the contemporary and the future simulation period. And so all of the output I'm going to be showing you are kind of from these 5-year simulations, but we really are only end up being interested in the first handful of years because oysters reach market size. But just to give you a sense here, these are a bunch of different simulations run under just in different environmental conditions in Chesapeake Bay. And so you can see that we sort of get the basic shell and tissue growth right. But what's interesting is that as the oysters sort of start entering, let's say year 2, year 3, the range and growth outcomes that just this model would predict from variability starts to get pretty big. So you end up getting a lot of variability in this. And we tried to capture as much of that as possible. And Matt will talk a little bit about how we use that variability to drive the economic model. Okay. So in the last handful of slides before I hand over to Matt, I just want to show some of the output and some of the main findings we found. And so we've done a lot of different simulations. We try to capture as much space as possible. And so there's sort of 2 model domains that we've used, and I'm really going to just focus on this one on the left. And so these are the stations that we'll be showing data from. And we really use these stations to try to capture as much space as possible in those places where we had high confidence in the model. Now, those of you that know the industry well will know that people aren't necessarily growing oysters out here in the main part of the bay. And so there's another half of this project that I can't really talk about today where we used data informed by model simulations in a lot of the shallow regions of the bay to estimate growth as well. So we're trying to capture kind of broad space and how oysters respond to different conditions in those different salinity regimes, but also trying to get close enough to shore to have a little bit better reality when it comes to where folks are actually growing oysters. So I put this slide together to summarize, the factors that we considered for oyster growth and what the modeled impact was, at least where we're at today in this process. So what you're going to look at is an environmental factor, the projected direction of change in that factor, and then how that transferred down to come to an impact on oyster growth. So if we start with temperature, it's said to warm, and our model mostly predicts that warming will be better for oysters. It sort of opens up their filtration window. But in some places it had a slight negative effect, primarily because it sort of added enough metabolic cost that they had some net energy balance deficits because of warming in the absence of otherwise good conditions. Salinity went up everywhere in the bay, and the way we modeled it, basically you start ingesting more and you're in a period where you ingest more and more commonly when salinity is high, so that was a positive effect. Acidification increased, more acidification, and that had a negative effect, but it was actually really small relative to the other factors we considered. And then food, which is really just chlorophyll, went up or down depending on where you were in the model, and it had a really strong positive impact, which is maybe not surprising, but it's the type of thing where depending on what the food conditions are in the future related to nutrient reductions or the effects of temperature on algae can have a, in our models that would have a really strong impact on what the outcomes were. And so I want to kind of touch on that food effect for a couple of slides. And so what I'm showing you here is all of the data for all 10 years we simulated for each site in the bay. So each dot represents a place for a particular year. And this is in the contemporary run. And you're looking at shell length at the end of 2 years versus this omega factor. And what you'll notice, which I think is telling, of all the stations we monitored, modeled, only a handful of them were really below 1.5 or 1 on average. And we wouldn't model much acidification effect below this. So a lot of them are up here in this territory. And if you look at what drives the variability in shell length on this axis, you can see that where you get the biggest growth is where you have the highest ingestion rates. And at the same, let's say, acidification, where you get the lowest growth is where you have the lowest ingestion. So you can really see the impact of food, at least in this type of model, and how it plays out. Another way to sort of summarize the future versus the past versus the future is to look at the shell length in the contemporary period that we modeled versus the shell length in the future. And this line here is the 1-to-1 line. And so where the dots fall below the 1-to-1, that means that growth was actually worse in the future. And where they're higher, it means that growth was better in the future. And you'll notice that wherever we saw better growth in the future tended to be where there was an increase in food energy. And wherever we had a decrease in the future, it was where there was kind of a decrease in food energy. So food sort of took on this really important role. So I want to just show you one slide that has details on this, just to sort of give you a sense for how this played out, and then I'll pass on to Matt. So what you're looking at here are some details of the model. I at least wanted to show a couple. And so we're looking at a station here that's kind of marginal, but there's a farm at this location. And so you're looking at kind of 5 years of simulation. And the black line is the contemporary period and the red line is the future period. So you can see this is a place where it shows a little bit of a decline in growth in the future. And one reason for that is that this red line here is that acidification tax, it gets a little bit bigger in the future. On the bottom left here is the soft tissue weight. So you can see that growth decline. But you can also see that this red line on the right indicates the smaller salinity tax, meaning that conditions were better for filtration as a function of salinity in this case. And then if we get to sort of the crux of the model, this net energy balance here is really how much net energy are they accumulating. And you'll see that it's down there in the summer that we see the net energy balance sort of get worse in the future. And part of that is due because temperatures are higher, but temperatures also offer a longer, larger filtration window. So there's sort of a balance there. And if we look at the ingestion rates, these are these two bizarrely named variables that are at the bottom here, we can see that there was less food at this particular site. And so this really sort of, I meant to show this to show some of the multivariate complexity of this, where, just if you have a place where it's predicted to have less food, you'll have less growth. And we can compare that, I'll just go through it quickly, with another station that had really not much change in growth in the future. And that was a place where food was more or less the same. It got warmer, but maybe a little too warm. And the acidification and salinity just weren't really a factor in the growth of the organism. So I'll wrap this up so Matt can go. But in conclusion, our model estimates today suggest that the bay will be warmer, saltier, and more acidic. And that the net effect of these changes is, at least where we looked, somewhat of a reduction in growth, but the availability of food that's projected, and we didn't even consider quality of food as a major result of that. The acidification effect as we modeled it was relatively small compared to some of these other factors, and that there's a lot that we can do in the future to get more refined model estimates, to improve our confidence in the bioenergetics, which is kind of a complicated thing to do, and start to look a little bit more in detail at farms and not just more broadly in the bay. So at that point, I'll pass it on to Matt and to the economic modeling part of this.
All right. Yeah, thank you, Jeremy. And before I dive in, I do want to just recognize that a lot of this analysis came out of discussion with the whole team. And in particular, I'd like to recognize Lisa Wanger, who's on the call, who worked on the economic modeling specifically with me. There's just a few things I'd like to go over. I'd like to give you a little flavor for the methodology, stochastic dynamic programming, that the economic model uses. I'd like to spend most of my time on the results, and then chat a little bit more about profitability thresholds, and then share some ideas for continued research with all these linked modeling aspects working together. Next one, Jeremy. So yeah, so stochastic dynamic programming, you haven't heard of this before, it's a bit of a mouthful. And if you aren't familiar with this technique, I would just think of this as optimization under uncertainty. Optimization gets a lot harder when you have probabilistic distributions. And here, the distributions that we're using are those for growth as well as for survival. And so direct solutions just become infeasible with all those things interacting. And basically, there are computer algorithms that just go through and search for what's the optimal strategy for each combination of state variables. That's what sets this technique apart. And those grower actions in this example, it would be grower actions that optimize the stream of net benefits over time. And next. Yeah, I wanted to put this up to just sort of summarize. I think of this as two of the foundational elements of the economics model. It's really built right on top of all of the ecological modeling and the biogeochemistry that Jeremy just described. And if you think of two of those legs that the economics model stands on as growth and survival, what we're looking at here are growth and survival variability across the bay in the current era, or historic era rather, and then the changes in those conditions in the future, with bottom culture on the left relying mainly on diploid stock, and then down the center, water column culture relying on triploid stock, so slightly different growth. But what's interesting to me is that not only does growth vary across the bay, that's sort of expected, but in the future, those future changes also vary across the bay. Not everybody experiences the same change in the future. Jeremy did mention some of that survival modeling, and at the end of the day, some of that low salinity potential mortality in the future. My understanding is that was more or less washed out by the salinity increase from sea level rise. And so we're going ahead without any change in survival modeled for the future. Next one. I did want to show the group just specifically the types of annual survivals that we're working with. Anything from the high 70s down to the low 60s. And these are from Maryland Department of Natural Resources, and that low salinity modeling was based on empirical relationship from Southworth et al. Next. Okay, yeah, and so now I think of this as the third leg of the economics model. We need to know something about grower costs, and there is information out there, and we do have separate models set up now for bottom culture versus water column culture, two very different ways of growing oysters. First uses diploid typically, the second triploid typically for faster growth, and different cost structures, different sales prices. So we've integrated all of that, and but really the main point for this presentation is how much we've adapted that to deal with uncertainty. So at each time step, your growth could be higher or lower than you thought it would be, and survival could be higher or lower than average. And so the model costs will flex according to that. If you have an especially low crop, you'll have lower costs because you'll have fewer oysters to husband and that sort of thing. Okay, next. Here's just a basic diagram of how this model is put together. You've got these state variables, and the model really runs on the growth draw and the survival draw. These are probabilistic distributions, and the survival distributions are, again, from Maryland Department of Natural Resources. The growth distributions are based on the different year conditions of the 1990s, and then again in the 2060s. And then in the center there, in sort of the peach color, these are the two things that the model currently allows a grower to do to optimize. I'm getting a message that I should start my video. Oh, okay, sure. Yeah. Okay. And so the two things that grower can do is optimize the planting density for their site, both their where they are in space and also what temporal area they're in, but also mitigation. Now, this is a pretty powerful variable. This is anything that can change or adjust either growth or survival in the future, or rather in any time period that you're in. And so those costs all go into net profit, and those shell lengths and your number of survivors in your cohort, those transition into the next year time step. I did want to mention that this is all coded in MDP Solve, which is a free software. I think it got cut off there, but thanks to the developer, Paul Fackler, for putting that out there. And this is running in MATLAB. I think there's also an R version for anyone interested. Next. Okay, so on to high-level results. And if it looks like it could be difficult to grow oysters at a profit here, that's my impression as well. This is bottom culture as well as water column culture, average net profits over time for both historic and future eras. And we did expect to see it being unprofitable at the far north and also the far south. What we weren't sure about was what would happen in the middle. And so we do see variety there. The linked models together predict that some of these sites would operate at a perpetual loss, a slight loss. And this would be after paying manager salary as well as all of the labor and all those other costs. It looks to be a fairly good location further north in the bay, not the extreme north, but towards the north end. And then towards down there, a site that's named there is CB 61 and 64. Growth is especially good at that part of the bay, at least the models suggest. And there again, it's a bit better for aquaculture down in that area. Next. So this slide is basically just to convey that just like survival and growth are on probability distributions, net profit is also on a probability distribution. So if you're right at the cusp of being profitable, you will not always be profitable. And so even if you're in a pretty good location, you're going to suffer some difficult times. Next. Okay, looking at just the acidification angle there and comparing here for water column culture, the very last bar is gray. And that's meant to convey the future without acidification. So in other words, Jeremy gave me some altered futures. Okay, he gave me a future that's just the baseline future, of course, but then a future without the warming impact, and then a future without the acidification impact, and ran that through what's the impact on oyster growth. And then I ran that through the economics model, what's the impact on profitability. And so then at the end of the day, we can see here that that lack of acidification, in some sites, you do see a little bit of a boost in profitability, but often there's very little change. And there's a similar result for bottom culture, not shown here. Next. Okay, yeah, I have a couple more slides that just kind of give a flavor for some of the other things this model can do, the economics model. And so imagine contrasting two optimized solutions here, comparing CB43, this being a site further north in the bay, and CB61, a location further south in the bay, has stronger growth but lower survival. And imagine you're mismatching the optimized solutions. You're going forward with a solution optimized for a different location. And the second and fourth data series convey the degree to which you would lose profitability on average. And in this case, water column culture is more sensitive to that optimization. Next. Here's another example, and here we're imagining a hypothetical brood stock that would cost more, but it would have some sort of boost in survival. Let's say it's a 5% boost in survival. That would still be from a probability distribution, but on average, survival would be higher. For bottom culture, now where's the cost breakpoint for that? At what point would it become worth it? Where would that broodstock start to make sense for a grower? So it turns out that at about 2 times the cost of standard diploid stock, that you would start to see some sites using that new broodstock and making a bit more profit from that. And that usage goes up a bit as the cost falls. For water column culture, it turns out that that cost would have to be a bit cheaper. But these are the sorts of scenarios that you can run through the model and look at time varying and site-specific mitigations and see how those affect profitability. Next. Yeah, I did promise to spend just a little bit of time on thresholds specifically. So what we're looking at here is just directly that trade-off between survival and growth. And it turns out that not only does your profitability, and these threshold lines here, this is right at the cusp of profitability, dividing the unprofitable space on average from the profitable space on average. Not only does that depend on mean survival and mean growth, but it also depends on the dispersion in those 2 variables. So what I've plotted here is, let's say you were drawing from a distribution of a wider someplace where that growth dispersion is wider, someplace further south in the bay. And then all of a sudden you've got a different profitability threshold, and you could all of a sudden be in an unprofitable space versus a profitable space. And we did see this with climate change, that some of those sites further south, not only did the median growth bounce around, I saw some widening distributions of growth as well. And again, the economic model runs on probability distributions for anything that affects the distribution, either the change in mean or the change in standard deviation, that will end up having some sort of effect on profitability. There's a third series here that would apply for widening survival distribution. I didn't plot that here, but that's another interesting relationship to look at. This is for water column culture, this plot here, but there's a similar plot for bottom culture. Next slide, please. So just a few conclusions to wrap up. Quite a bit of profitability variability across space, and that makes sense, I think, especially, extreme north and extreme south. Those places were unprofitable, and then some varying conditions in the middle. And not as much variability through time as we thought we might see. Jeremy mentioned those offsetting impacts. And all that being said, there are ways in which optimization for both your site-specific conditions as well as your time-specific conditions, there are places in which that can adjust and increase your profitability on average, but you should expect varying conditions, because that's just the conditions that people face. Next slide. Yeah, for my final slide, I just wanted to kind of suggest some ways that we could move forward in the future here. Gets also some ideas from the group would be great. We could look at other ways of modeling climate variability and maybe take some cues from deep uncertainty. The second bullet there, I think this would be really interesting, looking at the different forecasts and how could a grower adjust if they knew something about what the future year would bring. Not only do they know their average probability distribution, but they know the probability distribution for next year's growth and next year's survival. They should be able to squeeze more profitability reacting to that information. How much more profitable could they be is a good question. There's a lot of mitigation sort of scenarios that we could run depending on the effectiveness of those mitigations and their cost. We could see when and where those make sense. based on these linked models. And last, of course, we could look at more sites. Jeremy mentioned possibly looking at shallow water sites, more typical of aquaculture, integrate any new data. But then again, there's just anything could have a probability distribution in this model, and we could run that through. So far, we've only included those uncertainties for growth and survival. Sales price has a probability distribution. All kinds of inputs have probability. And we could also look at shell quality. We talked about doing this at the beginning of the project and kind of had to pick our battles, but acidification will have some impact on shell quality as well. And when and where would that occur could be another direction. So that's the end of my part here.
That's the end of mine too. Just also wanted to mention that this project was primarily funded by the NOAA Ocean Acidification Program. So we thank them for their support. Happy to take questions.
Great. Yeah. Thanks to both of you for that great presentation. We do have some questions that have come in. I just want to remind our audience again, if you have any questions you'd like to ask, please type them in the Q&A box and I'll be sure to ask them to our speakers. Our first question here, Jeremy, you may be able to speak to this more, but it was asking if the abiotic factors in your model at any point get compared with field in situ sampling.
Yeah, if I understand that question correctly, yeah, the physical biogeochemical models, so like salinity, temperature, those we've validated all of those in the past. I didn't show those because we've kind of been working with these models for a decade or so now, but we do validate that well. The one thing I will say is that, you know, one of the reasons we didn't go all the way up into the tributaries, into the shallow waters was, you know, like the version of the model that we ended up using was kind of a main stem oriented model and doesn't generally do as well in those areas. So we didn't want to bring in some model uncertainty that we couldn't really not grasp, but model uncertainty that we knew existed, I should say. That's it.
Okay, great. So next question here, Jeremy, you had mentioned one of the limitations of the model was looking at larval effects. How important are larval effects for aquaculture if larvae are being grown in hatcheries, and is it possible to create more ideal situations if necessary in a hatchery environment?
Yeah, thank you for that question because it allows me to clarify. It's true that the larvae are not really that much of an issue in the aquaculture setting. They're more of an issue in the generation of them and the conditions that the hatcheries have to reckon with. And, you know, that's not really a place where I spend a lot of my research time so far, but I know that the capacity to buffer hatchery water to deal with carbonate chemistry is pretty well established at this point. What's harder is when it's something that's not carbonate chemistry actually that causes hatchery failures. We've seen that happen in Maryland, at least, where there's been big hatchery problems that weren't clearly associated with carbonate chemistry. So it could be any number of things that impact oysters. Thanks.
Yep. So another question here. You both may be able to speak to this, but Matt, you were talking about the various cost trade-offs. At any point in the future, will the kind of model results and potential strategies to compensate for any cost trade-offs be made available to growers so they could use that information to inform their decisions?
I think this is a question addressing something that we're actually still looking for information on, is different effectiveness and different costs of potential mitigations. Currently, we've got sort of a generic knob that you can turn, and I can run the model where there's some mitigation that affects at the planting time. It could also be during a grow out year. And I can just plug in what that average effectiveness is and what that cost will be. What we are missing currently, in my perspective, is what those mitigations really are. And so I think that's where research could be helpful. And what I can do is sort of outline the value of that research. At what point would that cost be worth it if you had to develop that new brood stock, or even just research a mitigation that growers are already using. And this maybe isn't widely known how this is a mitigation strategy. I don't know if that answers the question.
I think so. So another question I'll pose to both of you. Does this model also work for other oyster species, in particular C. gigas is one that was asked about?
I can, at least from the oyster modeling part of it, I mean, this is, all of these models we, what's the right word? We sort of craft them to be representative of a particular any particular organism's physiology. And so this model could be easily adapted to represent gigas, for example. But in its current form, it wouldn't particularly represent that species.
Yeah, on the economic side, so I mean, I think I would think of, and Jeremy, correct me if I'm wrong, there's kind of a calibration step for the oyster growth. We need to have that set. And then we'd also need we'd need as much information as possible on grower costs for that other oyster species, gigas, and also survival data. Sometimes you think all those things are easy to find, but sometimes they're not. So I'd like to sort of qualify the response, and that would be great and a great idea. It would be, I think, it would behoove us to see what's out there before we could make a reasonable attempt to do that. I think it would be possible.
Another question here. Does the model's estimates for survival take into account the periodic triploid mortality events that have been known to occur in late spring in Chesapeake Bay?
I'll try to answer that. Yeah. So what I know about estimating survival is very difficult to do. However, people have been doing these repeat bar surveys for years, and those are all based on, those are actually based on triploid stocks. So if there's something that affects, or did I say, hopefully I said if it's based on natural diploid stocks out there. So if there's something that specifically affects triploids, I don't think that would be especially well represented. But it would, if there's good information on how to adjust survivals for triploids, that would be great to include. So be great to get feedback on that if somebody knew of work that we could leverage in.
And what I can add to that is that, the way that we applied these empirical measurements of salinity-induced mortality was intended to maybe not capture, it could capture a seasonally based mortality event, or even a regular seasonally based mortality event if it was associated with salinity directly, right? So we started going down that path because right in the middle of our project, there was a massive precipitation event, I think a historic one in the mid-Atlantic. And some of the growers that we worked with were reporting 50% mortality, 25% zero growth, and then 25%, modest growth in their farms. And so we tried to capture that, and it uses the data in such a way that it would compute it over, an annual cycle. So it would capture a spring mortality event if it was associated with salinity. But, knowing nothing else about the regularity of the mortality you're talking about, it's harder to say.
Great, that actually answers the last question I had that was asking about how well kind of infrequent storm low salinity events were captured. So with that, that's all the questions that I see. Matt, did you want to pose any of your own questions or ask for feedback on anything before we wrap up?
Well, sure, but actually, I see a couple things. Maybe they're just sent to me, but there's a couple things in the chat, I guess, or Q&A, at least the view that I see. This one's for you, Jeremy. Anna Garcia Condo, I think. Thanks for the lecture. Abiotic factors plotted are compared with field sampling.
I think she answered.
Yeah, that was the first question I asked.
Okay, thank you. Yeah. Then I got a question about the different versions of this software. It's very powerful software that made this project possible, I think, in many ways. There is, I think the most built out version runs with MATLAB, and I know that there is also an R version. I can sort of point Anna to the website, the GitHub location where those are posted, or I think it's fairly easy to find, but I'm more than happy to follow up with her separately or whatnot. Okay, yeah, I think that I would ask, as someone who admires oyster growers and this hard work that they do that really adds a public service for all of us, and some of the questions got to this, but what are some things that, if you're an oyster grower, for example, does this sort of check out with what you know? Are people making a lot of profit, or does it look sort of marginal to me? That's what I've heard from other growers so far. Sort of eyes on the ground, kind of ground truthing, kind of insights. I think that would be valuable for me.
Matt, do you want to perhaps provide maybe your email so people could reach out to you there? So we only have a few minutes left here.
Sure. Yeah, I'll put that in the chat here.
Yeah, I'll do the same if people have more specific questions or other questions that they didn't feel comfortable asking the group. That's just hosts and panelists. Well, thank you again for hosting us. You're muted, Kirsten.
Yeah.
Great. Thank you guys so much for joining us today and sharing the great research that you're doing. I think the, the economic profitability and the impacts of these multi-stressors and things is real important as the aquaculture industry continues to grow on the East Coast. So super helpful, super interesting, and we'll help loop people into, if they have comments or conversations specifically for you, we'll help forward those along as well. So thank you. And this recording will also be posted on our website. I know we've had a couple questions about that today because people had some trouble getting into it, but midacan.org is where those will be, and it'll probably be up next week. So thanks again, and we look forward to seeing you at our next webinar on May 22nd.
Bye-bye.
Bye.